All right, hi everybody. Let's get into Unit 1, Chemistry of Life unit. And today we're going to start with 1.1, Intro to Biochemistry. We're going to talk about elements of life. By the end of the video today, you're going to be able to describe the composition of macromolecules required by living organisms and explain why certain elements are absolutely essential for life. All right, so before we dive in, I want you to think about this. What makes an atom the smallest unit that biochemists care about? All right. And share that with someone nearby. You should have mentioned that atoms are a building block. They make up everything. They're needed for chemical reactions and then determine properties of elements. All right. And so here you see the smallest unit of matter and its components. So for the basic structure, an atom has a nucleus, which is the dense center, containing both protons and neutrons. And then you have regions where electrons exist around the nucleus, and those are negatively charged. You have your subatomic particles, right? Your protons and neutrons and electrons. So protons are positively charged, and they're located in the nucleus, and neutrons are neutral and have no charge, right? They're also located in the nucleus. We already said that electrons are negatively charged and they're found in the orbitals around the nucleus. You can see them negatively charging around the orbitals. And so a key concept here is that each orbital has a different amount of energy associated with it. So the closer to the nucleus, means that you have a much lower energy, and then the farther you go out means that it has a much higher energy. Okay. All right, so let's talk about valence electrons. So these are the ones found in the outermost circle, right? The one we said that are the furthest away and have the highest energy. Why do they matter? It's mattered because it's used for making different types of bonds. And they're the ones that actually participate in chemical reactions. So valence electrons, the rule is that most atoms want 8 valence electrons because this makes them super stable. It's called the octet rate. And atoms are usually neutral, but sometimes atoms don't stay neutral. When they gain or lose electrons, they become ions. All right. So there's two types of ions. We have cations that have a positive charge. They have more protons than electrons, right? They're positively charged. They have more protons than electrons. And then we have anions, which have a negative charge and have more electrons than protons. And so way you can remember this is that cations, like the animal, a cat, cations are positively charged, like cats having paws. And cations have more protons than electrons, like we've said. That's why they're positively charged. We could talk about sodium and chlorine here. So remember, sodium has 11 protons and normally 11 electrons, but when it loses 1 electron, it has 11 protons and 10 electrons. That's why it's positive. You always see the Na+. All right, let's get into our elements of life, which are carbon, hydrogen, oxygen, and nitrogen. And a key way to remember this is memorizing the acronym CHON. And so basically, what you see most of is carbon, and it's the backbone of all organic molecules. It has 4 valence electrons, so it can form 4 bonds, making it very versatile. Hydrogen is everywhere in living things. It's in water, in all organic molecules, and it just forms 1 bond. Oxygen usually forms 2 bonds and is super electronegative. And we'll see why electronegativity matters. And nitrogen forms 3 bonds and is essential for proteins in DNA. Any other elements that you see them in trace amounts, small amounts, but they're super critical to maintain homeostasis, which is a balance in your body and in your cells, right? You could think of iron, which is hemoglobin, calcium, which is bones, phosphorus, which is in your DNA, magnesium, which is in chlorophyll, iodine, which is in your thyroid hormone. And so without these trace elements, organisms die quickly, right? Organisms meaning living things. And so let's break down each of these elements. So why is carbon important? So we can talk about the geochemical cycles. And so carbon is the basis of organic life forms, right? Inorganic carbon is fixed from the atmosphere by plants during photosynthesis. So it's incorporated into the carbohydrates of the plants. That's how they're stored. And they're the main source of biomass in ecosystems. All right. Organisms utilize carbon to produce every biomolecule. So we're going to get into the biomolecules in other videos, but the biomolecules are carbohydrates, proteins, nucleic acids, and lipids. So you need carbon to make all these biomolecules. When organisms die, they decomposers recycle the carbon back into the environment. And so a critical point to remember is that organisms in carbon depleted areas, so where there's not a lot of carbon, these organisms will die because they cannot make the necessary biological molecules that we mentioned above. And so that's why we say carbon is essential for life. All right, now let's move on to nitrogen. And here you can see that it says for the gains, and we'll get into that for a second. And so why is nitrogen for the gains, right? Why is it important for building muscle? Inorganic nitrogen is fixed from the atmosphere by bacteria and other decomposers and absorbed by plants to enter the food web. Organisms utilize nitrogen to produce proteins. That's why we're saying for the gains. And that includes for muscle proteins. And nucleic acids, which you see in your DNA and your RNA. Nitrogen is recycled into the environment by decomposers. And so a critical point to remember here is that organisms in nitrogen depleted areas will die because they can't make proteins or nucleic acid. All right. And so in the nitrogen cycle, most atmospheric nitrogen over here is unstable, unusable by organisms, but the special bacteria down here fix nitrogen from N2, which is the atmospheric gas, into ammonia and nitrates. All right. That plants can absorb. This nitrogen eventually becomes the protein in your muscles and the one found in your DNA and RNA in your cells. So same as before, without nitrogen, living things die. All right, let's get into phosphorus. Why life as we know wouldn't exist without phosphorus, right? So phosphorus is used to build nucleic acids and certain types of lipids, which are called phospholipids. And phospholipids are found in your cell membrane. And so the critical point to remember here is that organisms in phosphorus depleted areas will die because they cannot make nucleic acid or phospholipids. So here, unlike carbon and nitrogen, phosphorus has no atmospheric reservoir. You don't find it in the atmosphere. It comes from weathering rocks and is often the limiting nutrient in ecosystem. Without phosphorus, you couldn't make DNA or cell membranes as they are made up of phospholipids. All right, let's get into an example. So you're going to apply your knowledge to this real world scenario. And so this is an aquarium example. All right, so let's get into this question. So think to yourself, how are the chemicals secreted by the fish being used by plant in this aquarium? So how are the chemicals secreted by fish being used by plants in this aquarium? And so here you can see that there's exchanging of material between the organisms. The bacteria are the ones right here that are representing the rod shaped. And so go ahead and choose your best answer. So which of the following statements best describes how molecules released by fish become nutrients for plants? All right, I'll give you a second. And you should have chosen D. The ammonia molecules released by the fish are converted to bacteria by the bacteria to nitrates, which are used by the plants to make proteins and nucleic acids. A is wrong because bacteria doesn't convert carbon dioxide to oxygen atoms for making water. B is also wrong because fish doesn't release that. And C is wrong because nitrates aren't converted to CO2 for carbohydrates. All right. All right, let's get into electronegativity, and it's probably going to be the most important chemistry concept for biology. And so electronegativity measures how strongly atoms attract electrons to themselves. So it's the measurement of how much they will pull electrons towards themselves. So you could think of this like electron greed. The most electronegative elements are fluorine, oxygen, and nitrogen. And so remember that in order, because it'll help you predict molecular behavior throughout the course. So this is determined by how many electrons are in the valence shell. And so you have to remember that the rule is the closer to 8 electrons they have, the more electronegative the element is. All right, so again, the important elements are fluorine, oxygen, and nitrogen. And fluorine is the most electronegative element. It's not common in biology, but you should know it. Oxygen is the more electronegative than nitrogen, but less than fluorine. And finally, we have nitrogen, which is the least electronegative out of these three, but it's still very electronegative. So electronegativity differences between atoms create polar molecules, and polar molecules can hydrogen bond. So we're going to get into polarity in a second. I want to go over this trend. So as you go to the right of the periodic table, your electronegativity is going to increase. And then as you go down the periodic table, your electronegativity is going to decrease. So that's how you can predict electronegativity based on the periodic table. And so here you can see fluorine, oxygen, and nitrogen. And you can see that fluorine is most electronegative than oxygen, but oxygen is more electronegative than nitrogen. All right, sorry I got cut off, but we're going to move on to electropositivity, and this is the measurement of the ability of elements to donate electrons and form positive ions. And so these elements usually have 1 or 2 electrons in their valence shell. And so remember, this is the complete opposite of electronegativity, because electronegativity wants to attract these electrons. And again, the trend for electronegativity is as you go to the right, electronegativity is going to increase. So here you see fluorine is more electronegative than oxygen. Oxygen is more electronegative than nitrogen. But over here, it's the opposite. So as you go down, electropositivity is going to decrease. They're not willing to donate any electrons, right? They want to attract electrons instead. And so you could think about these as because they have 1 or 2 electrons, they're more willing to donate an electron to an element that wants to fulfill its octet role. All right. And so examples would include things like sodium, potassium, and calcium. All right, let's get into electrons and bonding. So this sets us up for understanding chemical bonding and how atoms connect to form molecules. And so the electronegativity differences between atoms determine what type of bonds they'll form. and how electrons will be shared or transferred, right? We said that they can either donate electrons or attract electrons. And so covalent bonds are the sharing of electron between atoms. So here you can see that they're sharing electrons, and energy is stored in covalent bonds and is released when the bond is broken. So you always see energy stored in these covalent bonds. All right, so now let's move on to ionic bonds. Ionic bonds is the transfer of valence electrons from a metal to a non-metal. And so here you can see how it's trying to fulfill the octet rule, right? So sodium has less electrons than chlorine. So this one's electropositive and this one's electronegative. And so chlorine will want to attract this electron, and sodium is going to do the opposite. It's going to want to donate this electron. And so in doing so, you can see that sodium now has a positive charge and chlorine has a negative charge, because sodium loses an electron and chlorine gains an electron. All right. And so ionic bonds are weaker than covalent bonds and will disassociate in water, so we don't see them a lot in biology because they're weaker bonds. All right, let's get into polarity, and this is basically the direct consequence of electronegativity. Electronegativity leads to one of the most important concepts, which is polarity. All right, and so polar molecules occur when there is an unequal sharing of electrons across a covalent bond. So here you could see a partial positive, and here you could see a partial negative. Here it's electron rich, and here it's electron poor, meaning it has less electrons. This one has more electrons. That's what it means by rich and poor. And so this happens when a very electronegative element is bonded to a very small or very electropositive element. And so over here, I want you to guess which one's electronegative and which one's electropositive, all right? All right, again, this one would be electropositive because it has less electrons and wants to donate to the fluorine, which is more electronegative. All right, and so key characteristics is that polar molecules have an overall neutral charge, but they have partial negative and partial positive charges on the poles. So this symbol right here means partial. All right. Electronegative elements will be partial negative, and they're going to pull electrons towards itself, right? We said that fluorine wants to take or pull the electrons from hydrogen to fulfill its octet rule. Electropositive elements will be partially positive because it has less electron density near itself, and it's going to want to donate an electron, right? The way I remember polar is like I think of poles, two opposite poles, one having a positive and one having a negative end, right? The way I memorize electronegative is I think that negative people take things, they want to take your energy. Electropositive, I think of positive people usually want to give, right? That's how I remember it, but you could think of something else. All right, and here's a video on the polarity of water, and we'll talk about water eventually, but here's a quick video if you want to preview that before we talk about polarity of water. And here you can see it's polar because it has a partial negative and a partial positive. All right, let's move on to hydrogen bonds. So now that we understand electronegativity, let's explore hydrogen bonds, one of the most important interactions. Why is it only oxygen, nitrogen, and fluorine? So remember that these are our electronegative champions. So these are the most electronegative elements, meaning that they're going to want to take electrons, right? And hydrogens are bonded to them, have a partial positive charge that can attract other electronegative atoms. So remember, hydrogen is electron poor, so it will be electropositive, meaning these are electronegative. So then these will have a partial negative charge, okay? And so opposite charges attract. That's why you always see a weak interaction between hydrogen and these three atoms. And this is what forms a hydrogen bond. All right, so here you can see hydrogen bonding with nitrogen. Here you can see hydrogen bonding with oxygen. All right, and you're always going to mostly see those attractions. All right, let's talk about bonds and molecular shape. So we said things bond together, but how is the bonding determining the overall shape? So you always want to remember that the structure, shape, and chemical properties of a molecule determines the function of that molecule. So the way things fold and the way things are shaped will determine what the function, what its job is going to be. It's going to show up over and over again when we talk about enzymes, DNA, cell membranes, everything we're going to talk about in biology. And so again, remember that the shape determines the function, or the way I remember is that the structure determines its function and bonding determines the shape. So, all right. So an example preview is that water's bent shape, it's not linear, is crucial for its polarity, which enables hydrogen bonding, which creates all of water's life-supporting property, right? Changing the shape will make it lose its function. So that's why it's not linear, it's bent. All right, let's move on to the laws of conservation. So before we move to water in the next video, let's establish 3 fundamental laws that govern all of chemistry. First off, energy is always conserved in a reaction. So energy that appears lost is probably given off as heat. So remember that energy cannot be created or destroyed, only converted, and this is why our body generates heat during metabolism. The amount and types of atoms are conserved in a reaction. So for example, if you start with 2 oxygens and 4 hydrogens, you must end with the same amount, 2 oxygens and 4 hydrogens. I'm sure you guys have done this when balancing equations. You always end up with the same amount of atoms you started with. Atoms are rearranged. Again, they're not created or destroyed. And that's, again, why we balance out equations. All right, and so the third one is that the amount of bonds is conserved in a chemical reaction. So if you start off with 4 bonds, you'll end up with 4 bonds. So if you start off with 4 covalent bonds, you'll end with 4 covalent bonds. Bonds are broken and reformed, but the total number stays constant. And so this helps us predict reaction products. All right, so let's summarize. Atoms are the smallest stable unit of matter with element-specific properties. Nucleus contain protons, which are positively charged, and neutrons, which are neutral, and electrons, which are negative in the orbitals. Valence electrons determine bonding behavior. Most want 8 to fulfill the octet rule. Ions form when you gain or lose an electron. So, for example, cations are positively charged, anions are negatively charged. Remember the acronym CHON, carbon, hydrogen, oxygen, nitrogen, which are elements that are essential for life, each with specific bonding patterns. And you'll also find trace elements of other things that are critical despite it being in small amounts. Then we talked about like the cycles and how things are neither created nor destroyed. They just reworked and rebonded. And then we talked about electronegativity and how electronegativity predicts molecular interactions and polarity, right? All right, thanks for listening.